Hi everyone. Today I will be doing a quick overview of epistasis and more specifically single dominant epistasis. Epistasis is when the expression of alleles at a locus masks or modifies the expression of alleles at a different locus. So the type of epistasis I will be focusing on today is single dominant epistasis. For this, I will be using an example of fur pigmentation in rabbits. So for the purposes of this example, the white or lack of pigmentation is going to have the genotypes containing the dominant W or the uppercase W. The black will contain completely recessive all lowercase genotypes of lowercase ww, and the gray will have the genotype that is recessive for W, so little ww, but contain at least one dominant allele for B. So that'll be either big B, or big B, little b. So in this case, the dominant W allele is epistatic to the dominant and recessive Bs. Okay. So here is an illustration of the pathway. Here we have the colorless precursor for the rabbit to be white. It has to have the W dominant allele. So if the W allele, the dominant W allele is present, we cannot continue down the pathway. Now, if we don't have that and it's both little ww, it can produce the enzyme 1, resulting in the black intermediate. Now here is when we can move on to the B allele. So we can either stay stuck here or produce enzyme 2, resulting in the gray product. In order to be phenotypically gray, however, there must be at least one dominant B allele. And here is the dihybrid cross showing the single dominance. Here, two heterozygous white rabbits are crossed, resulting in a 12:3:1 phenotypic ratio. Here we are able to figure out the phenotypes by referring back to which genotypes code for which phenotypes. And here is the breakdown of that. Another cross we can look at is one between a white rabbit, big W, little b, and a gray rabbit, little w, big B, little b. In this case, the phenotypic ratio is 8:4:4, and here is a breakdown of those genotypes to phenotypes. And here are my references, and thanks for watching.